right, everybody. Well, thank you very much for the invitation to present today at this symposium and talk about some challenges and opportunities that we see for rendered byproducts in the circular bioeconomy. And out of this talk, I would like you to, we'll talk about what is a circularity and what are some definitions for sustainability that have been presented in the past. What is that sustainability? What are some challenges that are inherent to use animal byproducts in livestock diets? But also, what are some of those other opportunities that are many of those that we've known for a long time? Sometimes we forget, or sometimes that priority seems to be minimized by society. And I'll leave you finally with some tools for you to use. But the biggest message that I see that the pressure for environmental footprint of food production, even though food may be less than what used to be estimated, we all want to say we want to do our part for improving environmental sustainability or for decreasing environmental impact. So even though that the magnitude may be less than we have thought in the past, we still need to make some efforts to make contributions. The second element is that in many of the calculations of sustainability or circularity, a lot of those systems have been measured or developed in industrial ecology for processes that are chemicals or fuels or products. But animals are living systems that are very dynamic, and the response of the animals to the nutrient is very dynamic. Today, we saw the presentation from Dr. Guiyao Wu, and I think that the knowledge of functional amino acid will continue to evolve even after he retires. So animals are even very dynamic. So something that we know today will maybe change in the future because we'll know more about the response of the animal to the nutrients. And your business is, in some cases, supplying those nutrients to animals. And then, therefore, the best use of products is not only when they replace corn and soy in a diet formulation, when they supply energy or amino acids, but actually if they supply some physiological function that the animal needs. Because in a lot of the calculations for life cycle analysis, we talk about pounds of pork being produced, but we tend to overlook that the animal needs those nutrients for health functions. And every time we decrease mortality or every time we supply something that the animal physiological needs, we're doing a benefit to the system that goes beyond from what we can explain by energy and amino acids. So I think this one has been talked before, so I'll go very quickly, but every time I ask my students, so I teach a class and to all the students, I ask, what is your motivation to come to my class? And they will say, a lot of them will say, I want to know about sustainability because I want to eat more plant-based proteins, because plant-based proteins will save the planet. And boy, I spend a semester with those students, I ask the same question again, and the answer is the same. So sometimes those knowledge or those paradigms are so ingrained that it's not very easy to change. But every time we talk about animal production, we still think very linearly. And changing that to the circular system sounds easy, but not everybody does that so easily. As I was mentioning, some of these parameters for circularity have been developed by industrial ecology. So there is guidance from ISO or the ISO guidelines for circularity. And yes, those ones are for decrease waste or conserve resources or increase the value of the resource. So if we think, for example, as a chair, as a bioproduct, so chairs is a biomass, right? It's wood. And how much wood do I conserve in the creation of that chair seem to be relatively straightforward. But in the case of an animal, the animal is giving us a product that is not meat, right? The animal is giving us something else that goes beyond meat. Because if we look at, so the Roundtable for Sustainable Beef, the National Pork Board, dairy producers or egg producers, they all have in their sustainability goals, multiple goals, not only decreasing GHG, for example, but decreasing antibiotic use or improving worker safety and benefits. So many of these sustainability goals are multi-dimensional. Number one, so GHG is important, but it's not the be-all, and we need to make sure that we are doing progress towards many of these goals. Sometimes we'll see trade-off between making benefits in one goal to the other. But the second portion of this that is important is our goal is not to produce pork for the sake of producing pork. Although I like pork and I like how it tastes, so I'll buy pork for the taste of pork. But biological, we are buying pork because pork provides us amino acids or 4 sources of nutrients. And like Dr. Gu has said earlier, plant-based proteins cannot be compared in the same way as animal-based proteins. So if I want to compare an animal-based protein or a plant-based protein, let's say I feed a person, and then that protein will go to the mouth, to the small intestine, where it will be degraded by enzymes, and then in the large intestine, bacteria will modify that protein into other products. So for a long time, the quality of proteins were compared in a system called PDCAS. And PDCAS used to be done feeding rats because it's cheaper and more flexible to feed rats than humans to collect fecal samples. So if I feed a rat with 100 grams of protein, and then I find 20 grams of protein in the feces, I will say there was 80% digestibility. Good so far? Okay, 80% digestibility, total tract digestibility, then by an index multiplied by the concentration of amino acids, it used to be the system called PDCAS. Well, if anybody is a swine nutritionist or a poultry nutritionist or aquaculture nutritionist here, you wouldn't feed an animal based on this number, right? Because you know there is a mistake there, because the large intestine is changing or increasing the digestibility in a way that is hard to predict. What really the animal use for synthesis of protein and growth, what the bird will use for synthesis of breast protein, or the pig will use for growing the loin, is actually amino acids that are digested and absorbed in the small intestine, right? And so at the University of Illinois, when I used to be a student, my advisor always is cannulating pigs, putting a cannula in the distal ileum and collect ileal digesta, right? And now, let's say we fed 100 grams to the animal, we find 25 grams in the ileal digesta, therefore 75 disappear between the mouth and that end of the small intestine, and therefore we assume that 75% was digestible. Good so far? Okay, and that parameter we call standardized ileal digestibility, right? And if you look at a book called the National Research Council, or NRC, actually we're working on that newer version of the book, and we most likely are going to present the value of protein quality among feed ingredients based on standardized ileal digestible amino acids. And actually now the FAO a few years ago sat together with a group of experts and said, well, how we are going to compare, how we're going to move from PDCAS that we know is an imperfect system to a better system. Well, let's move to the dietary indispensable amino acid score, or dietary indispensable amino acid score, or DIAS score. So this DIAS score is a still imperfect, but much better way of measuring the quality of protein. And the reason I want you to know more about this, because our goal as people, professionals working in agriculture or working in the sector of animal production, is not just to produce meat. Every time GSGs are calculated per pound of something, right? Per pound of food, per pound of, let's say, meat versus per pound of soy product. Well, we are making an inherent problem there or a mistake because we are not calculating the quality of that protein. Make sense so far? So actually, then we should be looking at how well we are at producing digestible nutrients for humans, right? There may be other things that animal products are supplying that are not completely inherent in plant-based proteins, but at least digestible amino acids is an improvement from previous calculations. And as previously we said, there is a lot of resources from soil or from grass that animals that we don't feed directly to humans. And there are byproducts from the production of from food that we use to produce animals. And then there is this portion of the circularity that we were talking about. Well, the second element that I want to tell you is that the response of animals to the supply of nutrients is also very dynamic. And we think that we need to continue to make progress in this direction because it will help us to understand better what does the animal really need. In the past, when nutrients requirements were looked for animals, we were used what we call a broken line analysis. In this analysis, we feed the animal with increasing levels, let's say, of protein or lysine, and then that broiler will increase the breast size or the loin size of the pig, and then until a point where it plateaus, we don't see more effect of that, and then where it breaks that line, we call a broken line analysis. This, for many years, used to be the way that we nutritionists used to tell how much does the animal need to consume, use a mathematical model called broken line analysis that came from a conceptual realization that beyond this point, we don't see a benefit. However, sometimes those mathematical models is our conception forcing the math. My postdoc, one of the postdocs working in my laboratory, Dr. Ramirez Camba, and his former advisor, Dr. Krista Levesque, have come with a different model called the log linear model. And they suggest that where we see an increase in the concentration of amino acid, that response is more curvilinear. So there is information in the response of animals to the nutrients that is still important for us to know. And this is still speculative, but they still suggest that at the optimal protein retention, we see an improvement in protein retention at a smaller level than what we see optimal physiological concentration of the amino acids. So sometimes we need to feed increasing amount of an amino acid to get potential better physiological function. And this is an example of where we have seen this data starting to appear across the literature. So Christian took around 28 studies from the literature and then made a database. And within that database, he looked at remember this number that I said, this name, standardized ileal digestibility. So the intake of standardized ileal digestible lysine in gestating sows. And we tend to work in my lab a lot with soy nutrition, so we continue to look in sows. And the total number of pigs born. And he observed that, yes, that response is actually curvilinear. And in a way in which at low concentrations, he may see some increase in litter size, and perhaps the mother prioritizing the growth or the number of the offspring. An intermediate level, he'll see a decrease in the total number of pigs born. And then as as those experiments, he observes a higher level of lysine being fed to the sows, then he'll see an optimization of both the mother and the offspring. So some of this has been previously described in a life history theory is a theory that comes from ecology, where animals adapt to the provision of nutrients into the environment. But the point is that we want to have data analysis or ways to do mathematical modeling. Nutrients are the response of animals for those nutrients is dynamic. So what we know today has been changing. And the reason is that we now have more computational power than we used to have in the past. When I was, my advisor used to be a student, he will use cards in SAS with holes, with punch holes, and then put it in the machine, come next morning and see the results, right? And good luck if you made a mistake punching the holes, right? I'm too young to have seen that, but it was always explained that at the University of Illinois, there was a building, a room where people were doing that. Well, nowadays, we have computers with much more power, so maybe we need to continue to do that computational power to understand the biology in a better way. So nutrients in the animal are dynamic. And the reason I'm continue to say this about in the context of circular economy is because what we supply to the animal, what you as renders may supply to us nutritionists, is something that may have a value that over time we also will be changing because we will understand more of what the animal is needing. So now many colleagues of mine, we talk about multi-objective feed formulation, where maybe the formulation is not only in optimizing the diet for the provision of amino acids or phosphorus, but also for optimization of environmental impact or decreasing, minimizing that environmental impact based on life cycle assessment databases. Well, if we do that, I think there was a lot of discussion before of what constitutes a byproduct, what constitutes a co-product. One of my colleagues at the University of Minnesota, for a while, he came with the idea, okay, if something's going to the landfill, I will call it waste. If it's not going to the landfill, but somebody's been using it for something, then I may call it a byproduct. So when he calculated the percent of food waste, for example, that we have in North America, he was referring at the percent of food that goes into landfill. And that number continues to be high, and it will be useful to continue to decrease that number. So what I want to bring with this is that the discussion of what constitutes a byproduct and a co-product is something that sometimes we put category to something that sometimes is continuous variable, because value is something that is continuous, right? Something can be value 1 cent, 2 cents, 3 cents, but at some point we make a category, right? Well, in the US, we have about 40%. This is in dairy cows of co-products that are being fed to cows. Again, how we define those co-products will still be to evolve or will need to be revising that. But the point is that we'll like and we'll hopefully need to do more of that. Well, long time ago, we did some work with on rendered byproducts, and many of you know this figure very well. There is no one single rendered byproduct, but multiple rendered byproducts. And the nutrient composition of those rendered byproducts change by the different products, right? And so whether I have meat and bone meal, blood meal, or chicken byproduct, the concentration of nutrients varies. And to some nutritionists, variation is something a little bit uncomfortable because I've been in several sessions here during the conference, and many of my colleagues are saying a lot of the time that I spend is to make sure that the feed mill produces the feed at the right time, right? So when we have a lot of variation, that creates additional work for me and manpower that I don't have time to do. Make sense? And actually, the livestock industry, we have evolved a little bit of something that people have called like just in time, right? Some of our feed mills do not have a lot of capacity to, or our farms, to contain feed. And if somebody makes a mistake, oh, we still have a week before that feed is over. No, sometimes it's you didn't call on Friday to call for the feed. On Monday, pigs are without feed. Make sense so far? So I think variation has created a little bit of push back from nutritionists to say, oh, I don't want to deal with that because I'm so busy with something else. We've created mathematical equations to help them predict what will be the concentration of energy, for example, in animal byproducts that you are going to select. So if you analyze and look at the concentration of protein, fat, and make a calculation, you can know how much energy can there be in that byproduct for feeding the animal. So even though there is variation, well, but there are ways to cope with that variation. And we have similar equations, for example, to calculate the concentration of digestible phosphorus. And I want to make sure that I emphasize the importance, for example, of phosphorus. Have anybody heard of a term called peak phosphorus? Around the world, our capacity to extract and use phosphorus in an economic way may be decreasing over time because there are mines where we go get phosphorus, right? But then that part of that phosphorus go to crops or to the soil, and our ability in the future to recover that phosphorus is going to decrease. Make sense? So there is pressure not only on decreasing GHGs for the sake of decreasing GHG, but for example, to make more efficient use of phosphorus because of the eutrophication that phosphorus causes itself. But at some point, we're going to run out of easy extractable phosphorus. It's not going to be tomorrow. It's not going to be my daughter's problem, but maybe my grandchildren's problem. Therefore, to make it more sustainable, we need to continue to make resource use more effective. Well, but somebody told me that nothing comes free in life, and many of things that are useful may also have some challenges. So lipids are good sources of energy for animals, excellent source of energy, but also essential fatty acids, or like linoleic or linolenic acid. But those fatty acids are prone to oxidation during processing and during heat treatment. And the beauty of this is that those fatty acids are so diverse, and the products from the oxidation are very diverse as well. We do know that in this case, a measurement of oxidation is T-bars. It's not the best measurement, but it's one that is being commonly reported in the literature. And we did a meta-analysis where we look across multiple experiments, what was the response of pigs, and we have data on broilers as well, to the concentration of T-bars in the diet. And we see that that is almost linear. So for every intake of feed that has high levels of T-bars, we see a decrease in animal performance. Well, I know that this slide is quite complicated, but I want to bring to you the idea that amino acids are also needed to cope with oxidative stress. We have observed in this experiment, we fed oxidized oil to pigs, and we saw that those animals that were consuming oxidized oil will increase in the catabolism of an amino acid called tryptophan in a pathway called the kynurenine pathway. We did that by using LCMS, or liquid chromatography mass spectrometry, where we collect tissues from those animals. We analyze the small molecules that those amino acids are transformed to, and then we build back like a puzzle where we build a picture, right? And this is the final picture that we built out of that puzzle. The point that I want to make is that as we were talking about that nutrient requirements or supply of nutrients to animals are dynamic. If an animal is in low oxidative stress, maybe he doesn't need as much tryptophan. But if oxidative stress increases due to the provision of oxidized or rancid oil, then that requirement of tryptophan may come up. Make sense so far? So things change over time. Well, lipids are oxidized, but protein also happens to be oxidized. And oxidation of protein, like lipids, is also complex, that is oxidized in multiple products. One measurement of protein oxidation is called protein carbonyl. So if we account for the carbonyl group in proteins, we have some idea of how much oxidation there is in the product. So we compare soybean meal with meat and bone meal and poultry byproducts, and effectively we see that these heat-treated products that are heat-treated actually in the presence of lipids then have also higher concentration of protein that has been oxidized that we are measuring as protein carbonyl. And the p-anisidine value is another measurement for the oxidation of lipids. So whenever we heat proteins in the presence of lipids at high temperatures, we're more likely developing damage to that protein. We used to talk about Maillard reaction, but protein carbonyl seems to be a second or another way that we damage proteins. And it's important because then those damaged proteins will tend to increase the oxidative stress in the animal and change the use of animals in the performance of the animals. But we continue to have multiple sources of food waste that we can use or food sources that we can feed to animals. Yes, some of those can be oxidized, but they are an excellent source of nutrients. And we can know when we have analyzed that those sources of animal or waste byproducts have high concentration of energy and high concentration of digestible amino acids and phosphorus. So maybe we need to continue to look for sources of byproducts then to incorporate in animal diets, and those will be potentially coming from other sources of nutrients like food waste. Now, as I asked earlier today to the previous presenter, maybe we cannot feed that food waste directly to animals. We know that has high concentration of energy, high concentration of amino acids, but there are other risks of the transmission of infectious diseases. So using insect, for example, as a biological step in between may be helpful. Every time we look at life cycle assessment, so at the University of Minnesota, we have models to calculate environmental impact, and we calculate that environmental impact of producing corn, soybeans, and other feed ingredients that are used in feeding animals at the county level by a mathematical model and the combination of an economic model. And so we did the calculations of what would be the environmental impact in this case of terms of global warming potential of GHG for feeding pigs, a corn soybean meal diet with distillers dry grains with soluble, with thermally processed food waste, or with synthetic amino acids. And yes, feeding pigs with the thermally processed food waste in our calculations decreases the environmental impact. And actually, in this case, we were not using the models, the newer models with circular economy, is potential or the circular economy, this number can be even smaller. But again, as I was saying, one of the impacts, for example, or the challenges is with foreign animal diseases. Many of you may not be aware, but in North America, we have an outbreak of a disease called porcine epidemic diarrhea virus. And that one gave a bad taste in their mouth to many veterinarians, and it was very impactful. And during that outbreak, many veterinarians sampled feed meals, farms, feed, and found that there was virus in places that they were not expecting, like in the offices of feed mill, in the booths of feed mill workers, in feed ingredients. And potentially, that concentration of virus in the environment may have been the one spreading the disease. Well, there was an outbreak of African swine fever in China, in Vietnam. Many of the smaller farmers in those countries used to feed swill feeding or go to a restaurant, collect food waste, bring back home, and feed that to the animals. And that practice of swill feeding was really associated to the transmission of African swine fever and the rapid spread of the disease in those two countries. So swill feeding practice was one problem. And sometimes it's hard for veterinarians and many professionals in the industry to distinguish between swill feeding and byproduct feeding, right? As much as we can try to clarify to them, it's still not completely clear. And it's a work that we need to continue to do. Well, the way we do that is by doing actually risk assessment models and helping them understand that there are processes, and during that process, we can decrease that impact of the virus transmission. So actually, yesterday, my colleague Fernando San Pedro presented the symposium of American Feed Industry Association. He presented this work. As we said before, spray drying porcine plasma has beneficial effects in the diet of the animal. For example, in Europe, especially in China, there is the desire of decreasing the level of pharmacological zinc in the diets of young animals. So if you take a young pig, is drinking milk with the mother, you move it to another farm where now you're going to be feeding that solid feed, a lot of those animals get scours, right? But we know that zinc, a trace mineral, if we feed it at 3,000 ppm, the requirement is around 150. If I go 10 times that level, we see a decrease in scours, right? Well, but that zinc that later will go to manure will accumulate in the soil and eventually decrease yield of crops. So Europe said, no, what I mean, you cannot use 3,000 or 2,000 ppm zinc in the diets of animals anymore. You can only use 150 ppm. That's all you can do. Well, in this case, spray-dried porcine plasma can provide the young animal a source of protein with a biological value that decreases diarrhea, decreases scours. And that has been demonstrated by the nutrition community for years. And it's the point that I want to make. The value is not always on GSG. There are values on other ways to enhance animal welfare and animal well-being. At the end, we're still working with animals. So in our case, then we took the process. In the case, one of the plasma producers that we were working with, they were evaluating UV light irradiation, and then we used that into our calculations as well as baseline calculations. We calculate what in food safety is called a performance objective. And a performance objective will be the idea that how many batches of feed can I produce without having virus particle, and how can I account for all the steps in heat treatment or virus reduction that can kill the virus during the process. Well, these are the results of some of our calculations. I will not have time to go over all those calculations, but I think it's useful for many animal byproducts to have these calculations done. In a pre-outbreak scenario, so let's say we have an outbreak of African swine fever in a country, there is a period of time where farmers may sell animals to the packing plant that are excreting virus. Sometimes because the virus will not be, or the disease will be not noticeable, because they are asymptomatic, or sometimes because the farmer is making a rational decision. If the animals are going to be condemned, better I sell what I can now where I can get some money, because if I don't have the opportunity to get paid for those animals, I better try to sell them, right? So there may be cases where we see virus being in the plasma of the raw plasma. After UV irradiation, we calculate that there is about 6.8 log reduction of the concentration of virus. Well, during spray drying, there is concentration. So we are removing water. The solid portion is concentrated. So therefore, you see a positive number here, an increase in the concentration of virus by 0.5 log. Okay. Then spray drying decreases the concentration of virus by 4.1. And there is heat storage. Heat storage, the products are put into warehouses, and we calculate a 7 log reduction. Totally, we estimated that there is a 16.8 log reduction, And if our target is to have at least 7.3, means that 100% of the batches that I produce are meeting that performance objective or are deemed to be without virus. And so that comes to the management of that risk. Yes, African swine fever and other viruses are a problem to the industry. We can mitigate that risk by using processes and calculating that risk. We also, but we don't expect that all your plants are going to be, for example, putting African swine fever to test that virus inactivation. Well, in the food industry, surrogates are used typically to understand the inactivation of other pathogens that we cannot put in a food plant, like Listeria or norovirus. So many people have used surrogate. At the University of Minnesota, one of my colleagues used to be or works for many years with a virus called Emiliana Huslevi virus. Emiliana Huslevi is actually an algae that is in the ocean, and this virus is a megavirus that infects only Emiliana Huslevi, that algae. But that virus has a structure that is similar to that of African swine fever, and better yet, has a sensitivity to temperature inactivation that we've demonstrated that is similar to that of African swine fever. Good so far? Well, when we collect viruses and when we will look at after we process, let's say you spray dry or you render a product, you look at how much virus can there be in that product. Most of the time you will be analyzing for the DNA concentration. Okay, but the virus to infect a cell will need to be intact. If I find the DNA, I may not know whether the virus is infectious or non-infectious. So we also develop a procedure to understand whether the virus is infectious, and then now we can tell, is there virus there? And it's likely that that virus can infect an animal. And we call this one the RISC-NA assay, in which we have been evaluating sources of feed ingredients and what is the survival of the virus in those feed ingredients. So the point that I want to make is that all human activities have some inherent risk. If I want to change the light bulb at my house and I get into a ladder, I have a risk, right, of falling off the ladder. But there are ways that I can mitigate the risk, and there are ways that I can manage that and still get the benefit of the light in my house. What I'm trying to say is that of the human activities, rendering and feeding byproducts to pigs and chickens has an inherent risk, but also the benefit to society of that circular bioeconomy. And explaining that, I think, to society continues to be one of our challenges. I leave you here with some resources. We have a life cycle assessment database. We have a group of epidemiologists that have worked on the survival of viruses in feed ingredients. And if you would like to talk more with us, you're welcome to join us at the Minnesota Nutrition Conference in Mankato, Minnesota. So, take home. Environmental footprint of food production needs to decrease. Animals have a dynamic response to nutrients, and the best use of those byproducts is to find physiological benefits that help the animal cope with stress. The work that I present today cannot be possible without a team of collaborations and the economic support from many funding agencies. Thank you very much.